Next is autolysis. So autolysis is more commonly known as self-digestion, autolysis. So auto means self, lysis means to burst open cells. So it's self-digestion of your tissues. After death, autolysis starts when the cell metabolism stops. So general workings of the cell stop, and subsequently the integrity of all the tissues, of all the cells throughout the body are lost. So the cells just start to burst open, homeostasis is no longer maintained, things just start leaking out. So selective cell membrane permeability and active cell membrane transport ceases. So remember way back to your basic biology of cells, all that active versus passive transport, selective permeability, anything that requires energy stops. So you still get that diffusion, but everything starts to go towards equilibrium. You don't get anything packed into the cells using active transport anymore. None of those proteins that are used to transport across cells work any longer. Everything ceases. Okay. Autolysis resembles the active process of digestion of nutrients by live cells. So it's the exact same process. So it's as if your body is eating itself, or as the animal body is eating itself. However, the dead cells are not actively digesting themselves, as the common name seems to apply. They aren't using energy to facilitate digestion as they would as if they were actually actively digesting food, or if you're eating something. Instead, this digestive process is initiated by the cell's lysosomes. So once again, think back to biology. Those areas of digestive fluid used to digest nutrients, to get rid of waste products, lysosomes, they will release their digestive enzymes into the cytoplasm of the cell. This is because that active transport, or those active processes that keep those digestive enzymes contained, no longer apply, and it just sort of leaks out into the cytoplasm. So you get these autolytic changes. Now, these first autolytic changes begin in organs rich in enzymes, such as the pancreas, the stomach, and the liver. That's what you're seeing here. Pancreas, stomach, and liver just beginning that digestive, that autolysis of itself. Okay? Any digestive juices that are present at death begin to eat away at the organs. So the small intestine, you'll start to see digestion. The stomach, especially if there's a lot of acid, pepsinogen present in the stomach, you'll see that start to eat away. The mucosal lining of the digestive system is no longer produced and no longer protects the stomach or the small intestine or the large intestine. So you see all of this sort of stuff happening. So that's autolysis. Now, the eyes. The eyes tend to show the earliest changes after death. The very first thing you see is a cloudy film that will appear on the eyes within minutes of death, and the cornea of the eyes becomes cloudy within 24 hours post-mortem. Okay, so very, very early. If the eye is open and exposed to the air, occasionally a brown to black band may form on the sclera because of the drying of the eye. This is called tache noire. So tache noire is French, I think, for black spot. Okay, it's this black spot on the eye, and it may be mistaken as antemortem or perimortem injury because it looks like a bruise. So this bottom picture here, you see this tache noire along this eyeball in this human. That person died with their eyes slightly open. That sclera dried out. Now it looks like a bruise. For many times, that is mistaken by people who don't know decomposition as a bruise. Somebody was punched in the face. Okay, so that's tache noire. Now, the vitreous humor. The potassium concentration in the vitreous humor has been used to aid in postmortem interval determination just for years. The science behind this is that after death, autolysis causes the selective cell membrane permeability in the vitreous humor, and that active cell membrane transport, it just stops. So if you remember way back to your basic biology, your basic anatomy and physiology, this causes ions to diffuse across the membranes depending on gradients. The vitreous humor is more isolated than other structures. It's your eyeball, right? Okay, so it's more isolated than other structures in the body, and it's more resistant to bacterial degradation from decomposition, just because of all of those protective enzymes in your tears, your lysozymes, all that sort of stuff in your eyes. Therefore, it's a relatively stable little package of potassium. So the potassium gradient reverses that potassium gradient will move or reverse during the postmortem interval, and it diffuses from the lens and the retinal blood vessels into the vitreous humor. So you get this change in concentration of the potassium levels in the eyeball, just across this gradient. And what you do is you take a sample of the vitreous humor with a syringe. It's aspirated with a syringe. The sample is centrifuged, and the supernatant is tested. The relationship of potassium concentration and postmortem interval is linear, up to 120 hours. So that's actually a pretty long time that you can use this this information. It's a linear association between the concentration of potassium in the eyeball and time. Hooray. And it's described by this equation. So the postmortem interval here equals 5.26 times the potassium concentration minus 30.9. Neat. Okay, so this value is of most importance after 24 hours, within those first 24 hours after death, because other measurements are accurate in that PMI interval. So the algor mortis is most accurate within that first 24 hours, so that's usually what's used within 24 hours. Just after 24 hours, you move to this potassium concentration. All right. After 124 hours, that's when you get the equilibrium. That's when potassium concentration reaches its equilibrium state, and therefore it's no longer useful. So these levels, these potassium levels, are controlled by the rate of decomposition. So anything that affects this rate will also affect the potassium levels. So if you have for some reason, an accelerated decomposition rate, you want to keep that in mind, because that will affect this rate of potassium concentration as well. All right. Gastric emptying time. Gastric emptying time and gastric contents are helpful to narrow down the postmortem interval, naturally. If it is known what time an animal last ate, or what that animal last ate, it may be possible to use the information to figure out a postmortem interval. So the emptying time of the gastric or the GI tract is affected by many factors, things like solid or liquid food, the fat and caloric content of the food, water intake, volume of the food, so how much food is ingested, if an animal was fed meals, or if they had free choice, if they just sort of grazed along. It can be affected by the age and the size of the animal, although in cats, increasing age doesn't necessarily slow down the gastric emptying time. So you don't see that in cats so much that age affects it. Now it's best if this is placed in context with all other postmortem findings. So you look at gastric emptying time plus the ocular changes plus autolysis plus algor mortis, and then you can get a more complete picture. But in general, you got these general emptying times for dogs and cats. So for dogs, the solids will take 4.7 to 15 hours to empty, while liquids take much less time. Cats, same idea, 4.7 to 12.5 hours versus 1 hour. All right. Now, liver mortis. Liver mortis is also called hypostasis or lividity, and it's the pooling of blood due to gravity in dependent body sites after the heart stops beating. So the heart will keep the blood moving. As soon as it stops beating, the blood will pool and coagulate according to gravity. It is most useful in determining the position of the body at the time of death and whether or not that body was moved after death. So it's usually visible under light-colored skin. You can also see it in the mucosal lines, in the eyes, and on internal body surfaces and organs, especially the lungs. It can be mistaken for congestion on internal organs. So you want to be careful with this idea, You can see on the right-hand side here, 2 pigs showing obvious signs of lividity. You can see this. See this blanching right here. This is caused by compression of the capillaries, so the blood couldn't get in there. See lividity on here, and this is lividity on internal organs. So it's this darkened area here. These are the dependent areas of the internal organs. Now, the coloration of livor mortis is reddish purple to violet, but that may vary due to blood oxygen content or certain poisonings. Right? So cyanide poisoning, what else does that? Carbon monoxide poisoning, yeah, that sort of thing will change this color of blood due to that increased oxygenation of the hemoglobin. So you see that the change to cherry red in those cases. So any condition causing that increased oxygenation of hemoglobin, so that carbon monoxide, cyanide, hypothermia, just thought of that, we'll do that. That'll cause a bright cherry red livor mortis. Brown lividity, so a brownish coloration, is seen in deaths by potassium chlorate or nitrobenzene. So that causes brown lividity. Now, lividity may be visible from 30 minutes to 2 hours after death. So right around there, that's when you start to see lividity. It becomes fixed within 8 to 12 hours. So it's considered fixed when blood shifting and drainage no longer occurs because blood has leaked out of the vessels. So to determine fixation, all you need to do is you apply pressure to a livid area. If it blanches, if it turns white under where you applied that pressure, fixation has not occurred. If it does not turn white, lividity is fixed. Okay. If the body is moved prior to fixation, liver will shift, responding to gravity. So you'll see a slight discoloration from lividity in new dependent areas, and the presence of lividity or fixed lividity in non-dependent areas indicates that a body was moved well after death. After liver was fixed, that body was moved. So we see this a lot in, or not really a lot. We see this in, say, somebody died on their back, and then somebody moved them or turned them over after, say, 8 to 12 hours. In that case, you would see fixed liver all along the back surface and nothing along that ventral surface. That's a big indication of, hey, that person was flipped over after a while. Or if an animal or human dies in a hanging position, standing up, you get fixed liver in the dependent leg areas, in the other appendages, and then they were laid down. You will see no liver on their back, everything down at their feet. You just got to think about where would that blood pool due to gravity, and does it make sense given what I see in this animal? Okay. So areas that are pressed against a firm surface do not have lividity because of compression of the blood vessels, like I said about the blanching. This will allow a vet or a pathologist to determine if the body was moved and reconstruct the body's position after death. So once again, this pig, you can see these white areas on the pig. Let me point those out. All these little white areas right here. That's because this pig died on this side, and these white areas were pressed against the concrete. So this pig was flipped over after lividity was fixed. Now, the same applies for any constricting materials or devices on the body. This may reveal a pattern. So we see a lot of lividity in human deaths, or a lot of blanching due to elastic or belts in pants, constricted clothing, boots, anything like that causes constriction, which will not allow blood into those vessels. All right. So that's livor mortis. Next is rigor mortis. Rigor mortis is a post-mortem muscle contraction which immobilizes the joints of an animal or a body. And it occurs due to the depletion of ATP in the muscles. So once again, way back to your basic biology, that adenosine triphosphate, that energy that our cells use, once that depletes in the muscle proteins will stay locked together until decomposition breaks them down. So back to your basic physiology, the way that muscles contract, you got your muscle fibers that attach to each other, and it takes energy to relax muscles. This is why when you have cramps, you want to take in a lot of oxygen, or you've probably run out of ATP in those muscles. So that's what causes rigor mortis. Is ATP depletes, those muscles no longer have the energy in order to relax and to relax those bonds. Therefore, everything is locked in rigor. Now, rigor will start very, very quickly if there was post-mortem exercise or perimortem exercise. Post-mortem exercise, awesome. If there was perimortem exercise, violent struggle, maybe convulsions, or high body temperatures. So anything that can deplete ATP very, very quickly in muscles, if that happened right around death, then rigor mortis is going to set him very quickly after death. If rigor starts very quickly, it'll go very fast. It starts in all the muscles at the exact same time, but it is most noticeable in the smaller muscle groups. So first in the jaw and the eyelids. That's when you first see rigor. Then it moves on to those bigger muscle groups. Now, in general, rigor will appear 2 to 4 hours after death. It reaches full formation within 6 to 12 hours, and it lasts about 6 to 12 hours. It will completely disappear, so those cross bridges will completely decompose 36 hours after onset. So there's a rule of thumb. That's called the 12-12-12 rule of thumb. It takes 12 hours to fully appear, lasts 12 hours, and takes 12 hours to fully disappear. Of course, this depends on the cause of death, temperature conditions, perimortem factors, all manner of stuff, which is why it's that 12-12-12 rule of thumb. But in general, it'll take 12 hours to appear, last for 12 hours, and disappear after 12 hours. Okay.
All right. That's it for this video. You let me know if you have any questions.